Uh, Welcome everyone. Good evening. Uh, Welcome back to one more live session, and this is week 7 discussion. So the topics for today's discussion are amino acids, proteins, protein and proteomics, techniques to study proteome and proteome, techniques to study proteome and proteome part 2, and bioinformatics. So as like the earlier sessions, live sessions, we'll be discussing some sample questions and along with it some few topics related to it. And it will be a live session of 2 hours, but we'll have it in 40 minutes each, like 3 sessions, 40 minutes each. So kindly, once 40 minutes is over, kindly rejoin it in a minute, and we'll continue with the previous discussions. So feel free to interrupt me while discussions, and I expect your participation when I'm discussing the questions. And please do ask me questions and use. It'd be great if you unmute yourself and directly ask me the questions, or you can also post it in the chat. So we'll begin with today's session. Just let me know if my screen is visible and it's moving or not.
It's visible and moving.
It's fine, right? The question 1 is on display.
Yeah.
Okay.
It's visible.
Okay. So the first question is, it's about the amino acids. I hope you all have the basic idea about different amino acids, the polar, non-polar, aliphatic, aromatic, and their structures, and what is the main backbone of every structure, the optical properties. So let's discuss the different amino acids that are the questions related to amino acids first, and then we'll proceed accordingly with it. So I request you all to read the question that is given here, and we can proceed. Okay. So the first question says that except for one, all amino acids have the same general structure as seen in the graphic below. So that is the basic C group in the middle, the COH group attached to it, and then we have the amino group and the R group. So R group actually signifies the hydrocarbon chain or the side chain, which is different for every amino acids. This specifically this R group. Rest all amino acids have this particular structure C, COH, and NH2 is same. And as we know that carbon is capable of forming 4 bonds. So 4 arms of it, 3 arms, one is joined to the COH group, one NH2, one H, and another one goes to the R group. So this R group is the side chain group or the hydrocarbon chain, whatever you feel like describing it as. So now determine which amino acid is it. First question is this one. What structural benefit does it provide to the structure of a protein? Select the correct combination of from the following options. So I would like you to, as there is only one participant, if I'm not very wrong, so you can just have a guess and post your answer in the chat box, and then we can discuss that why your answer is correct or wrong, and what should be the correct one. So the structure here is very clear. There's one C group, a COH is attached to it. You say it's A. Okay, fine. Why is it A and not anything else? You need to justify your answer. Okay. So you may unmute yourself, or you can also type if you're comfortable in that. So how does proline look like? It's Navya, right? Okay. So Navya, how does proline look like? If I'm not very wrong, and I would love to hear your thoughts, but proline has an imidazole ring to it. So do you find any ring structure attached in this particular given structure? So keep the thumb rule of it is, whenever you're given an amino acid structure, there are some amino acids which have like structures at ring-like structures attached to it. Any standard book, if you go, you will see. Keep in mind straight structures which does not have. First, you have to keep in mind what are those. Like there's proline, there's histidine, there's phenylalanine, tryptophan, tyrosine. So 1, 2, 3, 4, 5. 5. 5 amino acids will have somewhat a ring-like structure attached to it. So whenever a structure is given, try to find that if that particular amino acid falls within it or not. Does it have a ring? Now, if doesn't have a ring, then just rule out the 5 amino acids. Now come to the basic structure. Like what did I say? All amino acids will have this part common. I hope my pointer is visible. This part will be common for all amino acids. This is the R group, it keeps on changing. And if nothing is mentioned, you don't know what R group is this exactly. So you will consider it as H. That is only a normal hydrogen atom. Okay. So now that you don't have anything, so the simplest structure of any amino acid is the structure of the glycine. Why? Because in place of this R, the R group here is only a hydrogen atom and nothing else. So, okay. So you just have one R attached to it, and that itself is a hydrogen. So looking at this structure of the amino acid that is given in the picture, this is glycine. And so what is the function of glycine? If we have an idea about helix, like alpha helix or the secondary structures of protein, we know that glycine has a tendency of destabilizing or breaking the helix. So now what does destabilizing or breaking the helix means? That it will not let a helix form, or it will destabilize a form structure. So why does this happen? Again, it happens because of the freedom of movement, or the conformation, or the structure you see is so small and the conformation is so simple in case of glycine. So the rotation of its bond and everything is very easy. So it can easily like break a bond and form with something else. That is why it is known as a helix breaker. So in case of a helix, what happens? A bond is formed between the CO and NH, between the N and N plus atom, N and N plus residue of a amino acid, right? Hi, Arushi. Okay. So between the C, I'll let you join. Okay, Arushi, we are discussing this particular question. There was one more student, but she left in between. So you can just read this question. I was actually explaining why the answer is glycine and what it does by meaning of helix breaker, hydrogen bonds in helix.
Okay, ma'am. I look into it now.
Yeah, you just have a read at it and then I'll continue. So I'll just brief the question to you, Arushi. So they've asked that this is the structure of amino acid, of any amino acid, and they've asked that what is the structure and what is the role of this particular amino acid. So we need to find the correct combination from the following options. So you can just have
It's glycine, ma'am.
Yes, you're absolutely correct. And why is it so? Like why glycine? What is it function?
Ma'am, you're not clear, ma'am.
Okay. So what is the function? Like it says that it hinders the formation of hydrogen bond in the helix, creating a break in the structure called helix breakers. So which bond is it breaking? Like hydrogen bond, I understand. The entire alpha helix is formed of hydrogen bonds. So which specific bond is it breaking? Like between which two residues? Do you have any idea?
I am not sure about it.
So have you heard about alpha helix? Do you know what it is?
Yes, ma'am.
Okay. So let's
It's a form of secondary amino acids.
yes. For not amino acids, it's like when many amino acids are joined together, it forms a peptide. Right. It's a peptide chain or a peptide. It forms a peptide. So that peptide assumes the structure of the alpha helix. Now the peptide cannot be elongated and just cannot it cannot exist right. Like it needs to be compact. So it is form of a secondary structure. What happens? The primary structure is just the arrangement or the just array of different amino acids together joined by the peptide bonds. Now they need to fold, not exactly fold, but they need to get uh, into a structure. They need to assume into a structure, right? So that is an alpha helix, and I'll just briefly discuss what an alpha helix is. Okay. So this is how an alpha helix looks like. If you can if you're able to see, so you can observe it from the bottom as well as from the above. So if you're observing it from the above, like in this picture, this is more of a cartoonish picture and this is from a standard textbook. So you see the boxes, the green, red, orange, these boxes are actually different amino acids, and they assume a they take the structure of a helix, and there's a backbone. Like you see, there's a backbone which runs in parallel to. Now the backbone is actually formed of bonds. Now what bonds? Between the CO and the NH. So you see, first residue, this one I'll just slightly elaborate. The first residue is CO bond, the CO group, the carboxyl group, forms a bond with the NH group of the fifth residues, that is N, 1, 2, 3, and 4. Between the N and N plus 4 residue of a peptide chain. And these hydrogen bonds actually stabilize the alpha helix. Now, suppose you have a structure here, this H. Now, this H is free to form bond with anyone and it doesn't form with this one. So, what will happen? This turn will not form, or the structure itself will not form exactly what a glycine does. It doesn't form this hydrogen bond. So, that makes it a hydrogen bond, that makes it a hydrogen bond breaker or a helix destabilizer. Okay. So why glycine? Because glycine have more conformational freedom than any residue. Because the side chain itself, you see all CONH groups are bonded here. And the side chain or the R group that remains in parallel with the backbone that I mentioned, and they remain outside actually. If you see these ones, the R groups are in purple. Now let's see the left, a more scientific and a standard picture. These R groups are protruding out from the helix. But you see the CO, that is gray and red balls, these balls, and the hydrogen, H and N, they are much ordered. So if this hydrogen bond is not formed between the CO and NH group, it will get distorted or the helix is not a stable one. That is what glycine have. Now glycine tends to make the structure more unfolded. What I mentioned that they need to get folded and be into a compact structure, but no, glycine is a bit rogue in this because glycine is kind of free and it has just one side chain attached to it, like just one atom in its side chain, not an entire, like a bigger one, like a CH3 or a CH2H5 group attached to it. So what it does, it favors to unfold the conformation by not forming the helix itself. Okay. On the other hand, proline is also a bond breaker, like a helix breaker. So because now with the concept, proline idea is different because proline has a imidazole ring to it and it itself forms bond, the hydrogen bond that it has to form with other residues that form with itself. So that is the reason why helix is. So I got this small idea from a paper which says that which amino acids have propensity of forming, which amino acid has propensity of forming the helix, or it can break the helix or form the helix, or it's some amino acids just are indifferent, like they don't matter. if they're present or absent, they can easily form a helix. So I guess these type of things kind of help you in your MCQ questions for different entrance examinations or even college examinations, where you just need to remember, okay, which one forms a helix. Sometimes we are not very sure of the concepts, we forget it, we often forget, but try to remember these two structures, that glycine and proline, they are not very favorable to helix bonds. They often tend to break them. So is my brief idea about helix breakers and alpha helix clear to all of to you?
Yes, ma'am.
Okay. So are you comfortable with the structures of amino acids and its classification and all of these?
You're asking whether I already know the structures, ma'am.
Yes.
I'm clear with some, ma'am, but not.
Yeah, so like a previous student who was there, so she by mistake told this is the structure of a histidine, the option one. So that is why, no, proline. She said it's proline because maybe two things can happen. She was thinking of the helix breaker as proline, but again, this combination was not right. So she was thinking of proline. So will the idea that the structure is proline ever cross your mind by looking at the structure?
No, ma'am.
No.
Actually, proline has a ring structure.
Yes. So there are 5 amino acids which have sort of a ring-like structure. So whenever we are given any structure to identify, we have to narrow down that no, if a ring is not there, then these 5 amino acids sure shot or not, because those are visible structures. Like you can easily differentiate or discriminate between the others. So I was just telling her that how to remember this. Okay, it's good that you know. That's fine. Okay. Now let's look into the third question, into my second question. So which is question 3, but anyway. So the question is, a peptide bond with very torsion angles designated A, B, C is seen in the diagram. So we need to identify them accurately. So Arushi, just give a try. Now first tell me, what is a torsion angle? First tell me this, that what is a torsion angle? We know what an angle is, right? How do we define an angle in general? When we say angle, what does that mean?
The distance between a molecule.
Angle. In terms of degree, you can say, but how do we define distance between 2 points as angle? No, that is not correct, I guess. Okay, anyway. So have you heard of the term dihedral angle or torsional angle?
No, ma'am.
Heard?
I'm new to this topic.
Okay. Very good. So let's discuss on this. So basically, when we discuss about angle between two planes, that is a torsional angle. Now I'll just go to my next. So this is one plane. You see the boxes. This is one plane, and this is one more plane. Now this is a 3D structure. It's not a 2D, but for observation purpose, this picture is done. So the angle between these two planes is a dihedral angle, okay, or a torsion angle. Now when it's a you see, it's again an amino acid. These are amino acid residues, and it is actually a peptide, right? Now when there's an angle of rotation between planes, that means there must be some restriction between these angles. It cannot freely rotate. Definitely it has rotating properties, but can it freely rotate between itself? No, there are some restrictions. So that is how it's decided that alpha helix, beta turns, are they capable of existing or not? Because there shouldn't be any steric hindrance. Now suppose this O is here. I'm just saying a random thing. It's nothing according to this one, like according to science. If a O is here and there's another H or another bond, another heavy atom here. So that will cause a steric hindrance, right? So it cannot be there. That is not feasible. Chemically, that is not feasible. So we have to see, this was actually decided by Ramachandran, and we are very, I don't know if you're aware, but you'll be aware in your future about the Ramachandran plot, which actually decides that every amino acid, every protein, in what form it can exist having suitable angles of rotation among them. So that there's no steric hindrance. And these angles are actually called phi, psi, and omega. So B is the answer. Now, how I remember this? So you understand this is a peptide bond, CONH. So the angle the peptide bond, the main angle between it, the C and N of the peptide bond, it is the omega angle. Okay. Between C and C is the psi, and here it's not written. The other one, A, is the phi. This one.
Yes, ma'am.
This one, okay. So N, and between the C alpha and N, it's the phi. Sorry. C alpha and C, it's the psi, and C and N, right? This one is the omega. So C alpha is the main central carbon atom, to which the first, actually, from the carboxyl terminal, if we see, it's that first one is the C alpha atom. So you understand that this is the R group, this is the NH group, and this is the COH group, right? And then again, there's one more NH group too. This is CN, like CONH group bond, actually. So the N and C alpha, now how I remember, these are very confusing. I used to get confused a lot during my turn. So mostly today we are talking about the tricks of solving these sums. I assume that you guys are a bit aware of the topic. So C and C. So using phonetics, I try to remember it's the psi. Omega, you will remember because the main peptide bond angle is the omega, that is CONH, CN. And the rest, what is remaining is the phi. So it is the C and the C alpha, between the C and C alpha. So that is the phi. Okay. Try to remember this structure. This is from Leninja. I will be sharing the links at the end of my slide. So is it clear which angles and how you identify them?
Yes, ma'am.
Okay. So moving to my next question. Okay. Just have a read and then we can discuss. So the question says that we have used size exclusion chromatography to purify a molecular weight, a protein which has molecular weight 352 kilodaltons. Next, you have to run the proteins. So I have discussed earlier, I don't know you were in that class or not, you were supposedly. What is a size exclusion chromatography? So when we have different sized proteins and our protein of interest, we know, okay, this has this size and we want to extract that or purify it among the complex mixture. So, or we don't know the size of our protein and we just want to see the different kind of proteins in it. What do? We use a size exclusion chromatography. So a size exclusion chromatography has different size porous beads to it. The smaller, so on the basis of that, it gets eluted and we collect it. The ones which don't fit into the pore will easily elute because it cannot go inside the pore, but the ones which can go inside, it takes time, so it elutes later. On that basis, we collect the different elutions in fractions and then we run SDS or a page to see what was the different molecular weights or what was the molecular weight of my sample mixture. So that is what a size exclusion chromatography does. On the basis of size, it resolves a complex mixture of proteins into its constituents.
Ma'am? Ma'am, in size exclusion, actually the heavier metals will come down first, ma'am.
Why?
Heavier, that is with more molecular weight.
Why?
Because they do not get into the beads, ma'am.
But if you are using something with a pore size which is exactly matches to that, then what will happen? And the rest don't match.
Okay.
That depends. Yes. So I'm not referring to heavy or small or big, not like that. I'm just talking about a technique. I don't want to get into that. You are correct in a way that if they don't get inside it, it will definitely elute first, and the smaller ones will go in. That is correct. But suppose you have similar size here and there, these type of different proteins, then what will happen? Whatever is the size of your porous bead, any proteins of that size will get into it and will elute later. And the ones which cannot get inside it will elute faster. That is the idea of it.
Okay, ma'am.
Okay, you are correct. You are absolutely correct in it. And so that is what we have done here. Because I just took it in that way, because here we know the molecular weight, right? We have used the size exclusion chromatography to purify a protein, which a molecular weight of 352 kilodalton. So we had an idea so that our protein will be of this size, and then we are running a page. So anyway, we have used beads of that. Okay. So the second part of the question is, now we are running it on page, that is polyacrylamide gel electrophoresis, one in presence of SDS, beta mercaptoethanol, which produce two distinct bands. And the molecular weights are 132 and 42 kilodaltons respectively. And also we have run one more page. 1 minute. Which is a native page. So I have a chat. Somebody is in the chat. Just 1 second. B. Okay. Let's see what the answer is. So from the gel picture, what is most likely has happened? Like, what has actually happened here? Anyone? Amardeep has an answer. Do you have an answer, Arushi? Now, first, let's discuss what is a page, what is a native page, and what is a page that has a SDS and BME added to it. So, what is SDS? In general, what is SDS and what is its role? I would like, just tell me this. What is SDS and what is 2-beta mercaptoethanol? What is it? Anyone? BME disrupts the disulfide bond. So, okay. So, if disulfide bonds are disrupted, what happens to the structure of a protein? My next question. So, in which level of protein structure disulfide bonds are formed? Primary, secondary, tertiary, or quaternary? My second question. Anyone? Tertiary. Very good. And Arushi, so what is SDS? What is the function of SDS in general? Okay, so let me discuss this. Okay. So Amardeep is absolutely correct. So he says that beta mercaptoethanol actually disrupts the disulfide bonds in tertiary structures, which very good. In tertiary structures, mostly it's intra-chain disulfide bonds with the chain. And in quaternary, what happens in quaternary structure? We have the interchain disulfide bonds. And what are these disulfide bonds and what do they actually do? So they form the tertiary structure and the quaternary structure. Now, if the quaternary structure breaks, then the protein is actually uh, coming down to its heterodimer or homodimer form or its subunits. Like one subunit or one dimer means that one subunit makes up one, it means that it's the one peptide chain. Now, if similar two kinds of peptide chain form together, they form a homodimer. And if two different types of uh, chain forms, two different peptide chains form a dimer, it's a heterodimer. So that is the concept of hetero and homodimer, and it comes in the quaternary structure. Now your quaternary structure is broken, so your peptide chains are actually different within the protein. So that when you are doing a page, you will have different bands for it, right? So for that, when you are using BME, that will happen. Now, what does SDS do? The function of SDS is to denature, that is to break the hydrogen bonds, to linearize the peptide chain or the entire protein so that it can render negative ions to a negative charge to it. That is the concept, that we make the protein completely negative and then we run it so that it runs towards the positive end, and that's the thing. That's what is happening. So negative to positive, and in that way, it elutes on the basis of its molecular weight. So when we are using that is SDS. Now native page is a non-denaturing. So in that we are not going to use SDS. So whatever is the native state, like its folded form, that structure you will get. Now that is what happens. You see in this native page, you have got one band which has which is a 350 okay 352 kilodalton, and here you have got two different bands. So looking at this picture, you see the homodimers or the heterodimers, whatever it could be, right? You are not very sure that it's a homodimer or heterodimer because it has formed, there's a one thick band at the same place. So you don't know. So this question is actually, answer is 4, that both B and C are possible. Why? The protein could be a homodimer or two heterodimers, one of 132 or 42. Two heterodimers could be of the same size, same, everything could be same. Maybe the amino acids are different and all, but it could be same. And also it could be a heterodimer, which is again made up of two homodimers, and again the size is so. This is not very conclusive from this picture that what exactly is happening here. Okay. So the question answer will be B or C to it. So is that clear?
Yes, ma'am.
Clear, right? So it's a function of SDS, beta mercaptoethanol, the concept behind denaturing and non-denaturing gel. Clear? Amar.
Yes, ma'am.
Okay. Okay. So I have taken a picture from the internet to just briefly say yes. So if we see this picture, now this is SDS page plus this is the denaturant or the beta mercaptan. So you see a native page has one band, means the entire unfolded or the native, the folded state is here. In SD, when you're doing just SDS, what happened is it becomes linear. The hydrogen bonds will break between the entire protein. So you see there are different peptides, just peptides. But when we are adding beta mercaptan or any kind of disulfide bond breaker, you see the homodimers and heterodimer. Like this was one uh, dimer or whatever it was. It was one subunit which got broken up into two. This was again one, and this is one. So that is what happens when you use different disulfide bond breakers too. So is it clear? Is it clear to all of you?
Yes, ma'am.
Okay. So here it's written, why does the larger protein show two peaks under reducing condition? So what is the reducing condition here? Anyone? One question is, what is the reducing condition here? And the second question is, why does this particular band have like two small bands here and these don't? So why is it so like that? Anyone? Okay. So reducing conditions, as I mentioned, it is the addition of any disulfide bond breaker. So what happens? A disulfide bond is formed between S and S. So under reducing conditions, it will get reduced. That is, it will turn to SH and SH, and the entire structure will like break. Okay. So that is why. And my second question, that this particular band corresponds to two bands here. That means it had two subunits. That is why it is resolved in this way when used a denaturant. And that is the reason. Okay. So that was my question here. Okay. Let's go to the next question. Okay. Uh, I request all of you to just have a read. It's a big question. Is the question clear to all of you? Okay. Let's go through the question together. Okay, we have just 1 minute 30 seconds left. Okay, maybe we can discuss the question and then discuss the answer in the next session. Okay, so so you have recently joined a lab that specializes in proteomics and infectious diseases. Your supervisor has requested you to construct a pipeline for your forthcoming investigations and incorporate both gel and mass spec based proteomics. What variables do you believe are necessary for designing a good proteomics experiment? Okay. So first thing is, we will discuss what proteomics is, one, and then now what is a pipeline? So pipeline is actually a flowchart of what the kind of work you will be doing, or in more refined way, it's the protocol of your entire experimental plan. So step by step, what you're going to do. And now we'll be discussing what is gel and mass MS based proteomics, and on that, then what are the different variables. So the different options are appropriate choice of disease and control groups. That now, because you're working with the infectious disease, we always do a differential work. So what is a differential approach? That is, one is will be a diseased cohort or a group of people, and one will be a same matched control. Suppose you are having a disease of age 18, you need to have a control group which will also be. Okay. Thank you for rejoining. Just let me know if my screen is visible once again. Oh. Is it, are the slides changing? Visible, ma'am. Slides are changing, right? Okay, thank you so much. Okay, so appropriate choice of disease and control group. So what does that mean? As I was saying, that you want to work on a particular disease, and for that you need to have your samples, or you need to have your subjects. So where will you get your subject samples from? Suppose it's a, you can identify the signatures of the disease from the blood, so you have to collect blood, and that will be from your subjects. Now your subjects will be of a particular age group, and exactly subjects of the same age group but non-diseased or who don't have a disease will be your control. Now, best quality of RNA primers. Okay, I would request. Muting. Okay, okay, yes. See, okay, Amardeep, let's just get through all the. So, third is high quality of trypsin. So, what does trypsin do? It cleaves a protein. And then purified TAC and PFU enzymes. These are used in case of, these are polymerases, which are used in PCR reactions or in polymerase chain reactions. Then sterilized room including HEPA filter. So yes, this is a mandatory for any kind of work, which so that we have a aseptic environment. And the sixth one is the purified antibodies and blotting kit if we are performing a Western blot or even an ELISA. So high resolution microscope combined with imaging source. Yes, so these are the basic requirements of a high-end lab. So now my first thing is that I will be doing gel based or MS based proteomics. Okay. And then I will be focusing on infectious diseases. So the very first thing is I need my appropriate choice of disease and control. One. Secondly, I am going to do a MS based proteomics. So I have to digest my samples, be it gel based or MS based. I have to digest my protein so that I can degrade it to, I can just go down to peptide and then identify which proteins are there in it. One. So high quality of trypsin enzyme is very much required. Then I need a sterilized room definitely, because it's again a infectious disease. So I will be exposed to both. I will be exposed and my environment will be exposed to unscreened blood or the blood which is known to have some infectious signatures in it. One. And then because I'm doing a gel based and a MS based, I will definitely be doing blotting and ELISA experiments, which will need purified antibodies and blotting kits. So yes, I guess Amardeep answered. His answer is correct, but I would like to briefly discuss proteomics with all of you. Firstly, because this is a emerging, this is a technique, or this is a era or a domain of science which has been explored for quite a while, and it is quite significant and substantial in terms of discoveries of biomarkers and of various aspects, not just biomarkers. And also, secondly, I specifically work with my fundamental tools and techniques that are all proteomic based. So I work on that, and that is why this is also my region of interest, and that is why we'll slightly discuss what proteomics is all about and how it helps us. So I would just give a very small example that what I work on, so you'll be able to understand that how it is, how I'm correlating proteomics with my. Suppose I don't know markers of a disease I want to find, or of a particular pathological condition, and I want to find which markers or which proteins are differentially expressed when a particular subject is having the disease. Okay. So either I can go for one marker, and one marker is always not a very significant, or will not give you a substantial result, like in terms of yes or no. If you have more markers or more signatures of a particular disease or a pathological condition, then you can be more sure that yes, this particular, if this particular subject has these markers in his blood or saliva, or could be anything, or plasma or serum, that means that yes, he or she is a diseased individual. Similarly, I work on aging. So I try to observe what are the different changes in the proteome of a particular model organism when that organism actually ages. So I use a Drosophila organism to see that as the Drosophila ages, what is the change in its proteome or in its whole, whatever protein it has, what are the different changes. So in different age group flies or Drosophila, I try to see that. And interestingly, I try to correlate, I use proteomics to see that in whole proteome level. And the most interesting part of it is I try to derive its correlation with circadian rhythms. So what we do in our lab is we try to see, we conjunct or we try to intersect these two things, circadian rhythms and different aspects that could actually be beneficial to humans, or in future it will be. We try to investigate or explore different aspects of health sciences and its correlation to circadian rhythm, along with using proteomics or omics based techniques. So that is what I do. So now let's get into it, a bit more details of it. So there are two types of proteomics. Like if you have to discuss proteomics, omics means whole and proteome means the protein. So omics is the expression of the in whole level. So it will be all proteins. In omics means all and expression. So an expression level, the entire proteome. Similarly, transcriptomics, genomics, interactomics, anything that means omic means the whole thing in expression. Okay. So what are the two different things, two different proteomics? One is top down and one is bottom up. So top down means starting from the cell. So in general, whatever top down and bottom up means literally in English, that is the same thing is happening, but you will see the process slightly different. So in top down, what happens is we take a cell, we take a tissue, and we extract the protein out of it, all the proteins that are present. Okay. Then we separate the proteins either through a SDS page, gel, that is a gel that it means. Now it is 1D, 2D, whatever it could be, or a LC, that is liquid chromatography, and then we directly inject the samples to mass spec and see the mass spec results, the MSMS, and the data analysis. So with that, we get the idea of different protein IDs, and also if the protein has some post-translational modifications. So that is mostly a quantitation. Like we can quantitate how many proteins are there, what are the different protein the post-translational modifications is then protein. So are you aware of post-translational modifications? Do you know what are post-translational modifications? Is post-translational modifications known to all of you? Okay. So because only one person has said yes, I assume rest of the two of you don't know. So whenever a protein is formed, that is it's translated, that is not the last point of formation of protein or synthesis of protein. It has to get modified by some modifications which we known as post-translational, which happens after the translation process is complete, or it actually happens in parallel. One is that, and so then it is added to it. Like it could be somolation, it could be the addition of CH3 group, acetylation, methylation. These all, so after the protein is formed, these groups are added to it, and then the final folding process of the complete folding that we mentioned, the native structure, it gets it into its native structure. So that is what happens. So in top down process, we take the entire protein, we directly separate it by gel LC, and then we do a mass spec. But in case of bottom up, what we do is we will also take the protein, we'll extract the protein, but first we are going to digest it to peptide level. We will use trypsin. So trypsin will chop the peptide bonds at certain positions, and then we'll get peptides. And then we will do a mass spec. First we'll do a LC, then we'll do a mass spec analysis. Now, after that we'll do after the mass spec analysis is done, generally we do a database search to see what we try to map the different peptides into a protein. So this will also help you give the PTM quantitation and a protein IDs of different things. Okay. So this is the most used, bottom up is the most used thing right now. So top down is also used. It's not that people are not doing, people definitely do that, but we also in lab, we are doing bottom up. We don't do in gel digestion, But right now we're doing in solution. Like we all extract proteins. I extract proteins from Drosophila and I digest it, and then I separate it by LC. Mostly I do a HPLC, and then directly I put it on to the. I give my samples for MS analysis, and then we have a different data analysis for it. So that is what happens in case of a top down and a bottom up proteomics. Okay. Okay. So as we were discussing, the question says what should be your pipeline or your protocol. So first is separation, digestion, then you have to detect, and then you have to identify. And then comes bioinformatics. So how we are going to like analyze. Now MS, it's an mass spec is an instrument. It will give you a raw file that needs to be analyzed using some softwares. There are Mascot, Sequest, Proteome Discoverer. We use it, and we can find the count of it, the IDs, as well as the different proteins that are part of it. So in separation, there are different methods. One is chromatography, that all of us know. It's mostly the HPLC that we do. It could be 1D gel, it could be this just one direction, that is 1D gel. Two direction, 2D, there's 2D gel in two directions. One in first on the basis of pH we separate, and then on the basis of the molecular weight, like SDs. And one is the solution, like direct solution. We trypsin digest it, one, or we can just directly put trypsin in the solution or the extracted proteins, then we do a mass spec. So that is how it happens. Okay, no, so now this is a paper that has actually been using proteomics to in the field of diabetes. So this is their protocol, or this is their pipeline. So they do two things. They could do a quantitative proteome profiling or a quantitative proteomics. So now the qualitative and quantitative. So quantitative is to quantify. Now the term quantitative, like to quantitate that how much of protein, what proteins, etc. are there, and qualitative means a differential analysis, yes or no, like absolute and relative it becomes again. So what is the quality or the in yes or no. So what they do is, they in case of qualitative, they perform gel based. They get the bands, they excise them in solution digestion, they have tryptic digestion of it, and then a mass spec, and then a data analysis. So this is your sample from what happens. And in case of quantitative proteomics, because we have to know exactly the number of proteins, and we label them. So if we are labeling them, it becomes much easier. So here we have a sample which is labeled. Here it's labeled. So now what we do is, we will definitely digest it. Digestion is always there. Digest it. Then there's a cleanup process, so that we get a purified, and we get the samples devoid of any kind of salts or any other impurities. There's a desalting process to it. And we clean the samples, and then we run a mass spec. So these are different labeling techniques: SILAC, ITRAC, TMT, LFQ. And LFQ is label free. That is also done. So I do this LFQ mostly. And so that's how you quantitate your proteins in it. So this is how a protocol or a pipeline of a, it could be infectious, it could be anything, of any kind of proteomics workflow look like. Okay. So is the idea of proteomics slightly clear to all of you in your level? Is it clear to you? Uh, yes. So, because you're saying yes, so have you heard of proteomics before, or have you seen anyone doing any proteomics technique in front of you? Have you have had that opportunity of observing? Maybe observing, not doing, I assume. So, Amardeep, okay. So let's get on to the next question. We have a chat. No, I had one subject on proteomics. Great. Okay. Means were demonstrated. Okay, okay, okay. Yes, yes. Okay, I understand. Okay. So let's move on to the next question and see what it means and what can be done. What could be the answer of it? Yes. So just have a read it too. Uh, do you know the question pattern? I'm sorry, Sharon, but I'm not aware of the question examination of the exam pattern. I'm really sorry. You can pose the same question to the discussion forum. Somebody will be uh, answering to you for sure. Okay. So, any idea what the answer will be? Or we will be discussing it definitely. Okay. So, let's discuss it. Okay. So, if I know you all have attended some lectures and you are aware of ESI, multi, you've heard these terms, but I guess you're not much accustomed to it. Otherwise, you would have answered it. So, if I show you this picture, let's break down the picture. What is there in the picture? So, somebody just tell me under the tag Maldi, if you're seeing this, what does this mean? Like, how do you interpret? If it's possible, like only three participants are there, I would highly recommend you all to unmute and just discuss with me in person. That would be great. We can just have a open forum and discuss that what this picture looks like. Okay, Sharon is right. Sample plus matrix placed on wells. So, what is a matrix? So, what does it mean? What is it made of? What do you mean by a matrix? Can anyone just tell me in one line what happens in a mass spec? We were discussing proteomics till now. So, what happens in a mass spec? A solvent which would transfer energy to the sample. Okay, right? You are correct. So, just tell me what happens inside a mass spec. So, what is the basic principle behind a mass spec? Okay, so let's go through this concept once again. So, what is a mass spec? So, mass spec is actually an analytical technique. We analyze it. Okay, in which what is done, in which molecules are converted into ions, which are separated on the basis of mass is to charge ratio. Proteins are converted into ion forms and their mass to charge ratio is correct. Correct. Is it correct? Yes. Exactly. Not just proteins, but any compounds or any molecules can be do. Like a lot of chemistry people do mass spec. So they are also doing mass spec. We are doing it in protein level, larger molecules. So it could be anything. So any molecule that is first converted into ions, then we are separating on the basis of mass to charge ratio. So when we are converting any sample into ions, what comes into my ionization? So we have to convert it into ion or make it ionizable. And so there are different techniques. The majorly two techniques. One is the ESI and one is the MALDI. So suppose you have a neutral molecule. It has no charge. Now you ionize it to either gain a positive charge or a negative charge. And these ions will be detected by a detector. And already your molecules have a mass. So on the basis of mass is to charge ratio, your ions are going to be detected or your samples are going to be detected. And then you have an analysis. So now, one method is known as the ESI or the electron spray ionization. So what is done here is first sample molecules are sprayed in an electric field to produce ions. So you mix your sample with a solvent, which could be methanol, acetic acid, acetonitrile, ammonium hydroxide like this, and also water to it. And then on the depending on the pH of the solution, the molecules will either have a positive or a negative charge. And then what you do, you spray it. You spray it. The solution now, the sample or the solution has your samples which are charged. Now you spray it through a syringe. It creates an aerosol because it's a high speed. From the tip of the syringe, this is what happens here. You see, this is what is happening in the ESI. So from here you are spraying and this is the sample. It has the solution in it, but this is a vacuum. You now will give a vacuum to it. Now this is the process which is known as nebulization. This formation of aerosol is known as the nebulization process. Now the vacuum is present in the chamber. So what will happen? Now you see the same circle is actually shrinking into small and finally it getting divided into indivisible ions, right? So what is happening? The sample is evaporating and only the molecules or your interest or your proteins or whatever it could be, they are just having the charges in here, correct? And they could be multiple charges, +1, +2, +3, like that in case of ESI. Now, the solvent molecules that have evaporated and you have the charge molecules behind and you pass it to the analyzer, which is in the right hand side of it. Now, to increase this effect of evaporation, a potential difference is applied to it. A strong electric field is applied to it, which is, then you see it's here, right? It's already vacuum and you're adding. So that makes the solvent molecules now will become electrically, that also becomes electrically charged. So now, because both the solvent molecules and your molecules, now obviously it will evaporate. It is not that it will not evaporate, but now the solvent molecules are also charged, your molecules are also charged. Now high chances they are going to repel each other. So this repulsion is actually causing the evaporation, right? So what is happening? You have a sample, you have so you are going to mix it with a solvent and you're going to pass it through a syringe like this, and it's going to form a what do you say, aerosol. So now the aerosol is charged because it is in and this entire process happens in presence of vacuum, and you are now applying what, electric field to it. Now because you are adding electric field to it and vacuum is there, electric field will create the solvents, will actually make the solvents also charged as well as your samples charged. Because they have disintegrated into individual charged entities, they will repel each other, and that repulsion will cause, along with the vacuum present, will cause evaporation of the solvents, and only your analytes which are charged will move on to the analyzer to be detected. So there they are going to detect it. Now there are different types of detector to it. We are not discussing it right now, but this is a part of the sample like the first how what happens when your sample is sprayed into a ESI or it is into an ESI. So is the concept of ESI clear here? That what is happening? I feel that if this is not clear, I can again like or we can go through this concept and okay, very good. And rest people, uh Arushi and okay, Arushi has left. So Amardeep, is it clear to you? Okay, very good. So now let's talk about MALDI. Now, what is MALDI? The definition or the full form of it is matrix assisted laser desorption ionization. Desorption it is. Okay. So what happens is, as you mentioned, there is some wells here. Okay. So, and there's something called the matrix. So matrix has small particles in it. So it is mixed with peptides. Now the samples are mixed with MALDI. Okay. And then these peptides are actually bombarded or subjected to laser. So what happens is this laser actually helps in the ionization process. Now why? The matrix molecule or the compound that was mixed with the peptides actually has a uh, conjugated pi system or a benzene ring attached with the OH group that is able to absorb the energy in the form of the UV light, that is the laser. So then which blasts the analyte into the gas phase, which is again charged. But keep in mind, in case of MALDI, it is just just one singly charged. Okay, it could be positive or negative, but it's always singly charged. It's not multiply charged like the use utility we have in case of ESI. So this is what is happening. And this process of when we are bombarding it with a laser and we have charged ions moving to the next phase of analysis, this is known as ablation. So that is the difference between MALDI and ESI. So I have just two small videos for you. Let's just see what happens in case of, okay. Before that, let's see the next part of MALDI and ESI. So, okay, here the desorption process of the crystallized matrix with analytes. If this picture is not clear, okay. So, field free drift, and then there's a detector. Okay, so here it is getting detected. What happens in case of ESI? There's something different known as a time of flight. So, just in case of SDS page, the heavier or the bigger molecules took time to resolve. They used to always be on above, and the smaller molecules move fast because their migrating capabilities are more, because they're small in size. Same thing happens here. If the mass is more, so MPIs that also will be more. So what happens is you see bigger and the more charged ones stays back, and the smaller ones will be actually detected before. So that is the same thing, time of flight. So the time taken for it to traverse the entire path till the detector is known as time of flight, and that is the principle behind ESI. So this is a time of flight. This is the electrical, more or less it's actually the same thing is happening here. So that is what here happens. Now if I increase this area, if this length is increased, so it will have more time to move and it is more resolved. So longer it is, the better. Okay. Is it clear? Now let's have a look at the videos. So what happens in case of matrix and analyte, how they are mixed here. Okay. So now first they're mixed, and then actually here also there's a process of evaporation. There's a step of evaporation. So what we do is sample has the solvent, and the solvent is allowed to evaporate from the sample. So actually it is a dried sample. Ultimately it will get dried. Then that is put into the mass spec, and again a vacuum is there. Okay. Again there's a vacuum, and so what happens is the target is or the sample is brought in the vacuum, then the wells are struck by UV laser, energy is absorbed by the matrix molecule, and they are ablated from the surface. This process is known as ablation. Like before we were talking about nebulization in case of ESI, but this process is known as ablation. It's ablated from the surface, and this carries the analyte molecule in the gas phase, that is the next phase. And from here, the matrix molecules that carry positive ion charge is generated. During ablation, the analyte molecules are ionized by proton transfer. Okay. Let's not get into so much of detailing. So that is that what happened, and let's see the process of ablation here. So this is the matrix that here the laser will hit it, and here is the sample and the matrix mist, and this is the plate. You see how they are disintegrating and like different colored in colors. So that is how it's, let's go through it once again. Yes. Okay. So is ESI and MALDI clear to you? Now let's go back to the question. Okay, now I've already shown you the answer. Now try to answer it now, once that we have discussed. And let me say, both of these are soft ionization techniques. They don't hamper or degrade the sample. So that is why they are known as soft ionization. Both of them are soft ionizations. So can I have one answer after the discussion? I'm giving you all the clues, right? It's a soft ionization. So just think that it, just just give it a thought to the rest of the questions, like which are just which don't have soft ionization, and just don't narrow it down and then see two, and you feel it's two and three. So converts analyte into liquid phase ions in ionization performs which of is it liquid phase ions in vacuum? Which one? And what is hard ionization? I just told that both of them are soft ionization techniques. There are no liquid phase ions. The samples are in liquid phase, which are ultimately evaporated in vacuum. So that doesn't make it a liquid phase ion. So both of them will be in gas phase. All are gas phase. Like there's no liquid in it. Only the part liquid is the sample, and then till the source of ESI. Whenever the spray is occurring, the liquid part gets over there. So let's see. Sorry, B. Oh, you need not be sorry. It will be B. So 1 and 3. Ah, 1 and 3. 3 is in multi analytes mixed into the substance. Where is my cursor? Yes, both of you are completely correct. So just remember, both multi and so ESI are soft ionization because they don't degrade samples. They have no effect on samples. Okay, and it converts into gas phase ions. What? Let's see the ESI. So this is a gas phase. The ultimate ions that are formed. What did I say? That they are completely evaporated. So there is no concept of liquids there. Liquids are not going to be here. In both cases, there is no liquid only. Here, to first only the evaporation takes place. Here it takes time. That a vacuum thing and the electric field comes into play. Then it gets evaporated. But both are in gas phase. Okay, clear? Okay. So this is slightly information based question. So just let's discuss what were the issues that doesn't. So in 2001, the first draft of the human genome was released, and the objective was to read the entire human genome from start to finish. So scientists from all across the globe confronted a number of challenges in sequencing the whole genome. Which of the phenomena does not describe the issues that have arisen from the Human Genome Project? So, any idea what the Human Genome Project was all about? or what was the aim of it, and why is it necessary? Why idea about the whole genome is very necessary? It could be human, it could be, okay, we'll discuss. Multiplication, replication starts, okay. Sequencing. Why is it necessary to know the entire genome? Why? What could be the reason behind it? Why do we need to know the entire genome? So that means yes, yes. Genome by genome, we mean the sequence. So why knowing the sequence? Now tell me, we are aware of viruses, bacteria. So we know the first thing that scientists, whenever something outbreaks or we come to know about the a new variant of a disease or of a bacteria or a virus, the first thing scientists do is they sequence it. So what is the sequence of any organism? So a sequence is an identity of the organism. So the sequence defines what is present within the organism, everything. Like from the sequence, we are able to know every small detail of it, like literally everything of it. So this was a huge milestone in biology of like sequencing the entire, like now we have this idea about the of what our entire sequence is made up of. So now if we know that what is the sequence made up of, we are able to design primers or biomarker discovery or any disease that is happening, that will be easy to know the cause if we know that all the genes present in a human. And also something is going to come up is people are working towards it. It's the personalized medicine, that is on the basis of your genome structure you will be prescribed medicines, which is again a part of gene therapy. If you sometime you have some disease which is a genetic disorder and it cannot be cured with normal medication, so and your gene needs to be targeted, so there's what your sequence will actually help you. It's not very easy and it's not also very economic to sequence here, because again sequencing is very costly. So now this human genome project were able to sequence the entire genome of a human. So let's directly go into the answer. The answer is E. So two things were not part of the issues that had arisen. One is sequencing multiple replication sites, one, and other one is sequencing non-coding genes. So these were the two things that could be done. So and and another option, shotgun approach. Using shotgun approach only, the entire sequence was sequenced and it is maintained in different libraries. So gene libraries, not like the libraries where we go to read books. So it's known as gene library, where we maintain part of a gene of the whole genome in different individual cells. It could be bacterial cells, it could be any kind of cell. So that is how actually a gene library is maintained. So you, whenever we want the entire genome, we take it and we try to join. So that is how it's done. It's another process of RDT. So yes, so it will be option A and C, that sequencing multiple replication sites, and C will be sequencing the non-coding sites. So is that clear? In a minute, the session will end. So I'll just wait for the session to end, then we can discuss the last part of. Thank you for rejoining. So we'll continue. Hmm. Okay. So are you aware of bioinformatics? Is my screen visible? Okay. So let us briefly discuss what is bioinformatics and what bioinformatics means and what is its role. Like if we break the word bio and informatics together. So because the next few questions will be slightly different from the normal questions that we discuss. It's more of informatics and uses of different tools and databases. Okay. So, what is bioinformatics? So, bio is the biology that we read, which includes molecular biology, chemistry, biochemistry, biophysics, then pharmaceutical sciences, different omics based studies. And if we join it together with the help of computer science and information technology, assisted with database management systems, computational resources, that that makes it bioinformatics. So because such a load of data is created, so we have so much of data that is generated from each experiment. Suppose be it a sequence of a gene, we have millions of strains of different organisms, there's so many species of it. So to maintain all these, we heavily require the help of informatics, so people who are really good in it or experts in this field. So they take our data, manage it, or analyze it. Now there's a part of data analysis after each experiment which use different mathematical expressions, statistical sciences. So using that we are able to hmm. So that is what bioinformatics is all about. When we are mixing these two, like these are the different things. So let's start discussing the questions on informatics and what it means. So the very first question is this, and I hope you have. Amardeep, is my screen visible to you? Okay, so he left again. No problem. So the first question that we are going to discuss here is I have included bioinformatics course in my curriculum. So in my first class, I got to know about NCPI. So NCPI is a website which is actually a database that stores different informations, and it has different applications and tools that we can use for our different purposes. So my first assignment is that I've given the accession number of a protein coding gene. So an accession number is an identity, or that I say in a very layman's term, it's the roll number of a particular gene. So it could be protein coding or it could not be. It could be. So the thing is that if I'm in a class, I am assigned a roll number so that I can be identified. So accession number is similarly an identity code for a particular gene. So that ID is given here. We have to enter its ID, and then let's see what kind of information we can retrieve from NCBI. So the first page that will open whenever you go to NCBI is this home page. So, so there you have a drop down menu which will have these many different options. In that you have to select gene, and then you have to paste that ID 3611 here, because we know that it's a protein coding gene. So under gene we can do that. And when we do so, this is what opens 3611 under gene. So this is what is given here. So the gene ID is 3611. You see, it's written updated 9th of March 2023. So databases are regularly updated because genes, informations, all these keep changing. And so they're regularly updated. Many a time, suppose I find 10 strains today of a particular species. Tomorrow, high chances I will find more 5 strains. So every day things are being discovered, and every day the database is updated. So this is what I find. So here the official signal is symbol of this gene is ILK. The name of the protein is integrin linked kinase, and it belongs to the Homo sapiens or humans. That is what it is. And you will have the ensemble ID to it. Type of gene, it's protein coding. Refsec, organism, lineage. So it belongs to Homo sapiens. So you see all from eukaryote starting to hominidae and Homo. And also, what are the different synonyms of these genes? You will also find and a brief summary of it, that what it does. So what I would do is we would go to NCBI, okay, and we will see how to explore this. Okay. Hmm. As I mentioned, we'll go to the drop down menu. We try it with gene, and we will just type it. Okay. So here we have this page. So I'll just slightly zoom in a bit. Yes. Yes. So we see ILK is the official symbol, integrin linked kinase, and this is how we actually perform it. And there are many things. The genomic context will come a bit later. That where is we can go to the genome data viewer. The view will, like, you'll be able to see which the different transcripts, the products. If we click onto this, or we just take the cursor here, this is the ILK gene, and you see the gene, what is the span, align length, serious length of it, the links to different tools, that links to different tools that you have this, and now you can go for further exploration. So you can keep on exploring these things. So yes, that is what we could do. Now I believe that because it's a protein coding gene, we can also try to search with the same ID in protein. Let's see what comes. It says it's a record removed. Okay, that's a different one coming here. Back to, okay, let's check once again. A different kind of results will come here. So yes, for this method, you should go to gene and then select 36.1 or whatever the accession ID is given, and then you'll find it. Yes. So you see the ID is given. So now let's get back to our slides. Hmm. Okay. Amardeep, it's clear how to, the basic of it, how to like explore NCBI. Let me know if you just want me to repeat the entire process once again. Okay, sure, sure, sure. So the question was that you are given a accession number of a protein coding gene. Suppose ID of a gene is 3611. So it's basically an identification code. Like, okay, this protein gene, like you are given a roll number in your class so that you can be identified among all your friends. It's not possible always for the authority to know that, okay, your name is Amardeep. You need an ID for it, which will be small. It could be 2-digit number, it could be a 1-digit number. Similarly, all the proteins, we cannot just remember it by name, or in database it cannot be entered just as names, right? So for that, what is done is they are given specific IDs to it, like identification codes, and that is a accession number in case of NCBI. So what is done is we generally need to just input this in NCBI and see what information we can have. So I was explaining, and these are my slides, you can check it later. So let's go back and see what is actually done here. So how do you find where is NCBI? You just write NCBI. Okay. National Center for Biotechnology Information. Click all databases. Now there's a drop down menu. You know that it's a protein coding gene. So you click gene and you paste the ID 3611. And you just give a search. See, you will have the entire details about this gene. So the name of the gene is integrin linked kinase. Symbol is ILK and it is in Homo sapiens. Now, if this gene was in mouse, it would have a different ID. We will see that also. So now what are these? This is a summary of everything, like what is the name, symbol, primary source, the different ensemble. Ensemble is one more database like that, different databases. It's type of a gene is a protein coding gene. It is present in Homo sapiens, and also the summary of what this gene does or what is, and in the below you have the genomic context, that which location on the chromosome it is, the assembly of it. These are all accessions, and like these are different numbers that indicate which location it is on, like that. And these are the different transcripts, the products of it. And yes, then RNA expression. These are different. You can play around with it. The pathways from PubChem, that which pathways this particular gene participates in it, like the reactome pathways or the now reactome KEC. These are again databases which actually have the pathways, like every gene, which pathway it is and how it is connected to other pathways. That is all. These are, I'm not going into details. Interactions, like a particular protein interacts with other proteins. Right? So which proteins it can interact with, these are present in here. You will actually be able to retrieve most of information in your level from it. Yes, the functions, geoterms, everything is there. So this is what happens when you search for a gene. Now I believe that we could search this for musculars. That is, we'll go to protein and search it. We'll just see that what comes. If we look at for other organisms, the gene ID will change, definitely will change. Same protein is present in others, you see. Animals, plants, fungi, protists, bacteria. You can find your species here. Here, if you see integrin linked kinase isoform A in Drosophila. Then this is yes, Egyptus. Other species, other mollusks, sorry. So rhinos in Rhinophlis. And okay. So if I just click on one example, maybe Drosophila. So you see the ID will change. Everything changes. Right. Yes. So this is what happens. Now let's go back to the main page. So this is a unique ID for for this gene. If you search with this ID, you will be like sent to this page. Isoform A is 2 times in Drosophila. Yes, because multiple isoforms are actually discovered. So you see here, both are you see both have same amino acids. Both are isoform A and E, but the accession number is different. The GI number is different. So that means that the sequence is different, the position is different, but both are isoform A's. They have been discovered separately. That is happens even in case of isoform B. You see, this is also different, but the accession numbers are different. So that matters. It could be the same protein, but the makeup of it is different. Somewhere the position or something or the other is different, but both are isoform. You can take any one whenever we are doing any kind of thing. You can just do a blast, or maybe we can get into the detail, the more further analysis when we. So that is what happens here. Now let's go to the next question. Okay, answer is there, but it's okay. Hmm. So the question is, in which chromosome number does this gene reside? Pick out the correct ensemble ID for the above mentioned gene. So now this is one gene. So we need to find in which chromosome it is, and we have to find the ensemble ID for it for the above mentioned gene. That is the same gene. So let's go back to this page. So it's saying on which chromosome number it is. So we first ensemble ID is clear here. That is this one. So you can just match it with it. I'll just share my screen like this so we can see. Yes. So this is the ensemble ID to it. You see, if you match it. Yes. And now we need to find out which chromosome number. So location. In location, you will find that location means in which chromosome number. That is the location of this particular gene. So this is which chromosome it is present. Okay. That is how you find the location and the chromosome number. Okay. Okay. Let's go to one more question. The question says, the above mentioned protein has various documented 3D structure available in PDB. This protein database bank. Now, PDB is one more database which has different proteins and the crystallized structure present. You will go and you will see how it looks. Please find the correct PDB ID for the same. What is the resolution of the mentioned document? And pick the correct option. So, what we do is, now PDB looks like this. It's again the homepage of PDB. So it's a protein database where till date there are this many structures and this many computed structure models. That is just computationally done, not experimentally discovered. So here we have to paste the name of the protein, or many a times there's a PDB ID, but now here we have to find the ID. So first we are going to copy paste the protein name, and then we are directed to this page. So here we have to select Homo sapiens. That we do, we select Homo sapiens here, and you see there are so many. It was said in the question that there will be many different like structures and many different IDs. We need to find the correct one. So how do we do that? Now we have to search. and then we will find this one. So let's do it. I will keep this here open. Just copy it. RCSB PDP. Like this, you go here. You paste it here. You specify the name and source of organism. It's in Homo sapiens. And then you click this on refinements. So now you see. So structural basis of competition between this and this binding to the integrin. Then we have 3FCQ crystal structure of integrin linked kinase. This will also not be. Now, structure of integrin kinase actually repeat domain in complex. So we have to have a structure which we need to have an ID which says the structure. Now let's see how it looks. Click on it. From here, you will get the resolution. Okay, at resolution at which it was discovered. So this can be moved, I guess. Yes. So you have all the different integrin, how many chains are there, like A chain, B chain, length of sequence, protein, of course, sequence length of protein, organisms, the different mutations, how it looks in image if I click. I don't know why the function is not working here, but you can actually rotate it. 3D rotation is possible here. The features, hydropathy index, everything you will be able to access from here. But according to the question, you just have it in. Now, expression system in E. coli. So when it was discovered, it was actually expressed in E. coli. Let me click on random things and let's explore this. 3D view. Okay, yes. So you see, you can move the protein and see. So can you identify the structures? What secondary structures are here, Amardeep? What is this? Can you identify the structure? How does this look? All the bonds and everything are being shown. So that is how we do. This is the alpha helix. And this is helical in nature, right? And this is the beta sheet. And these are the turns or the loops that we say. So that is how alpha, 6 alpha helixes and some beta sheets. Good, good, good. Very good, very good. So that is the beauty of these websites or these databases that you see, you're able to see the structure, you can get it. And now this is in a very preliminary stage that we use the structures for. Yes, we use the structures for defining all these, but in future you will be there are different methods of docking studies. There are different simulations. These structures, PDB structures, are highly acceptable and very much required. So that is how they serve the purpose. So I guess I'm done for today. Is there any question that you want to discuss, or you want me? Is there anything that is left, or you want to discuss anything? Uh, so I would end today's session, and we'll be Oh, thank you. Okay, yeah, Amriti. Thank you so much for joining and staying till the end. And so we'll be meeting next week again. That will be my last live session. And see you in the next session. And if you have any discussions regarding exam or anything, do post it in the discussion forum. So somebody or the other will answer to it for sure. Okay. Thank you so much. Thank you.